Good evening and welcome. This is the Turtle and Tortoise Preservation Group's a special live stream event, first of many that we have in store for this year, 2025. Hope the new year is treating everyone well. We have a great group of guests today, an amazing topic, an amazing journey that we're going to share. Well, actually, we're going to let the authors share with you. But before we go around, before we dive into the topic, I would like to take a moment to go around and introduce everyone. I'll actually let them introduce themselves. But let me start with one of my favorite people in the hobby and here at Turtle and Tortoise Preservation Group, James Badman. Good to see you again, James.
Good seeing you, Michael. Thanks for having me on again. It's great to be back.
I appreciate you, and I'm glad you could make hop on here. James is one of those people who knows this topic that we're talking about. He knows what is involved, and he will probably share some of his past experiences on that front. But James, quickly, 30 seconds for those who are not intimately familiar with you. Can we talk about your background? Can you introduce yourself?
Sure. Yeah, absolutely. I'm James Badman. I'm one of the directors with Turtle and Tortoise Preservation Group. For those who don't know me, I'm an associate director of animal care at Arizona State University, but also manage a rather large private collection, not just tortoises, but a lot of exotic animals as well. Love the kind of the breeding and management aspect of keeping animals and working with them, as well as a lot of the behavior work and stuff that we do or participate in on the research side. Fascinator, love this topic that we're going to talk about tonight. It's just such a great story, and really look forward to hearing it from the authors. So
awesome. Steve, we'll go right to you.
All right. Sure. So I think people probably haven't seen me in person before. Most people. I'm Steve Poterala. I am a engineer in my day job, engineering manager at a pretty decent sized company, and but I have been obsessed with, loved turtles and tortoises since I was a little kid in elementary school. And, I think a lot of people in the hobby are sort of like that. And it turned into sort of this lifelong thing. And, I got out of college, started, met my wife. And when I met her, I had a couple of tortoises, and it just sort of snowballed from there. And so I work with a bunch of different species, something like on the order of 20 species. Have always wanted to do more on the conservation side, on, contribute in some way to research in the field. And so this was just an interesting opportunity that came up to sort of be involved in this project.
Awesome. Paul, if I can have you introduce yourself.
Sure. Paul Rattay. I live in Sonoma, California. I've always been kind of a turtle hobbyist and aficionado. I've kept various turtles and tortoises since I was 10 years old, but never really participated in any big organizations. I know a lot of turtle people. I generally stay pretty much under the radar. I'm not usually on forums, and you don't see my name on classifieds a lot or anything like that. But every once in a while, you may. And then recently anointed citizen scientist with this topic that we're here to talk about tonight.
Awesome. Aaron, how about yourself?
Yeah, Aaron Johnson. I live in Missouri, not too far from Kansas City. And I began keeping tortoises in 2003, started with star tortoises of the Indian and Sri Lankan type. Very quickly moved to adding Chaco tortoises, hatched out my first Chacos in 2004, and pretty much stayed with them until 2021. And then I expanded out with some Burmese stars, Speaks Hingebacks, finally moved into the very difficult project of the Bell's Hingeback. Recently successfully bred some of them, successfully treated some for ventricular coccidiosis, and still working on that project, particularly the breeding. Canixi's homiana as well. And my background is in biology and biomedical sciences.
Awesome. And last but not the least, just in time to introduce himself, your friend from Libya. Let's hear from you.
Yes, hello everyone. How are you? So happy and so glad to be among you here tonight. To introduce myself, my name is Murad. I'm from Libya. As I mentioned before, for my friend's colleague, I'm a PhD student at nearest university north of Cyprus. My specialized area, environmental education management. I work with several environmental NGOs in Libya to protect wildlife and marine organisms, especially tortoises in Libya. These kind of species, it's endangered now in Libya because, as you know, we have economic and security problems. So that's why many of people make tortoises in illegal trade, like smuggling between the boarding between Libya and Egypt. So we have to make action plan as a Libya citizens. We have to make action plan to stop this smuggling and to protect this kind of species in Libya. Yes, sir.
Well, glad you could join us. Like James mentioned a minute ago, this is a story. Both he and I were hopping on a call and we were talking about this, and both of us were kind of getting excited because this is, again, like Paul mentioned, citizen scientists coming into play. We've talked about we're probably a bit biased, but we have often said that private keepers who do it with focus on paying attention to details in maintaining groups properly can do a lot of good for the animals, and can help our colleagues in the zoos and working in the field as well. And this is, I think, a perfect example. These gentlemen have worked together to describe a new subspecies of Greek tortoises, and we're going to hear from them about how this unfolded. So, Paul, Steve, Aaron, my first question to you is: where did this journey start, and how did it, unfold? Who wants to go?
Pointing at Paul.
Yeah. It definitely started with me. The journey started in June of 2021. When wow. I think going back to like 2017, I had always wanted to work with Libyan tortoises. I had always known about the Libyan Greek tortoise and thought it was like the most beautiful thing I've ever seen. And I think most of you and the audience know, like, for a period of time, and we just didn't see them, right? Like there were imports of those like decades ago, and very few of them had survived, over a period of time. 2021, I was out where Michael lives, traipsing through, some beautiful country. And of course, in a little bit of downtime, started looking at the classifieds just out of boredom, and saw, someone had posted these Libyan tortoises for sale. And I was like, wow, I never see those. So I just kind of immediately said, I'll take them, just kind of impulsively. The guy reached back out to me and just said, well, he had like 1.2 of them, like one male, 2 females. And he said, I actually have 2.4. Do you want all of them? And I said, yes, I'll take 2.4. Right. And that's kind of what started the journey, because when they arrived, 3 of them looked like the classic Libyan Greek tortoise, right? The Cyrenaica, the one that's very well known. Murad is intimately familiar with them because he's, done a lot of work with them in Eastern Libya. But the other 3 looked more like what, at the time, I was like, these look like Tunisian tortoises, right? Like that, just at a glance, and then, from there, everything kind of unfolded, right? Like the story definitely unfolds from there.
So you mentioned you're not in this field. You don't you kind of stay under the radar. So when you decide, okay, you got something that looks unique, something looks different, you start poking. How did this gang come into the fold?
How did like Murad?
Yeah, well,
Yeah.
Murad, Steve, I mean, you
Honestly,
started pulling people together.
I think for a few months, like from June 21 forward, it was just me. Like, and I was just kind of like, what do I do? I reached out to Chris Leone. I reached out to the Tortoise Trust. I had posted some pictures on the Tortoise Trust Facebook forum, and basically asked, Andy, like what is this, right? Because this clearly is not, just the typical Libyan tortoise. And most people were puzzled. Like everybody was looking at them and just saying they've got to be Nabulensis, they've got to be Tunisians. There's no other small tortoise that's that size that looks like that. Everybody started kind of slicing and dicing them morphologically. But then there was always like one characteristic that they're like, but that one characteristic doesn't really match. And you have three of them, and all three of them, have that characteristic. So people were like, I'm not sure that's what that is. So from there, I just kept, what I learned about myself, that I've probably already always known is when I get obsessed about a subject, I can go deep. And be tenacious about it, right? From there, just doing internet research led me to Egyptian forums and Libyan tortoise forum that was run by a man named Nasser Boussen, who lives in the UK. And Nasser was the one that said, I'm pretty sure that's a Libyan tortoise, and connected me with Murad, and basically said, you should speak to Murad. He's doing, he works with confiscations, relocating confiscations from traffickers, that are going through Libya on their way to Egypt. And, at some point I connected with Murad. I probably had posted some of this publicly enough that Aaron and I started talking about it. Aaron introduced me to Stephen and a lot of other characters. There are actually a lot of names that have like contributed to this. But that's kind of how we got to Murad and the Libya connection. And, without it, none of this could have happened, right?
Wow. It's a small world. It feels like a small world but gotta love it. So what was the light bulb moment where you guys kind of said, you know what, looks like we got a new subspecies that hasn't been described? What was that moment like? When did that happen?
I'll let Stephen answer that one.
Okay. Yep. So during this conversation, at some point or another, I'm not sure who turned us on to Gendica, the DNA lab in the Netherlands. And
that was what's his name? Boris Berlin in the Netherlands, I believe, or he might live in Germany. Okay. And he's the one that when I was talking to him, he said he mentioned on his Instagram that he got some confiscations and that he had them genetically tested, and that they were Moroccans. And then I had reached out to him at that point and just said, where do you get your DNA testing? Because I tried very hard to find a domestic DNA testing source, and it's just not available. Like most of them only will work with either institutions, schools, other labs and things like that. So I at one point, I think UC Davis offered it, but no longer offered it. And I ended up, at that point, we were introduced to Gendica and then started, sort of working with them for our DNA testing. Yeah.
So essentially we sent some samples to Gendica, and they, the very first samples came back and the reports were very confusing. They had made a typo in one of the initial reports that was sent back on what the subspecies was. And I think the report said it was, they gave it a letter haplotype of E. And then I think they called on one of them. They even called it Buxtonii or something. And we're all sitting around staring at this report, scratching our heads, and we're like, "What is this? Why? What is this?" And so then it becomes well, let's dig through the literature. We have to find every paper that's been written about this. We figure out. We go into Gendica. We pull the entire, or we go into not Gendica, GenBank. And we pull the entire GenBank library for Testudo graeca. And we start looking at every file. We figure out, well, what did they match this to? Okay, here's what they matched it to. Finally, after several rounds of digging, we find out that, oh, this has been published on before. The DNA card that it matched to was from a paper in 2017. But it wasn't in the paper. It was in the supplemental material for the paper, effectively, that in the entire study, they found, I think, a handful, but they only had one sequence in GenBank for a tortoise from a market in Libya. And it was not given a location. It was not given a name, but it was recognized all the way back in 2017 that there was a unique subspecies of tortoise somewhere in Libya. And so when we dug all this up and we're like, wow, this is that mystery tortoise that was found in 2017, and presumably they didn't have a location, they didn't have enough material to describe it. And then this is where the connections with Murad and the folks in Libya came into play. It was like, well, where did this thing come from? Where do we go look for this?
And that's kind of where I started becoming like super sleuth. I had reached out to one of my good friends. You may know Jim Buzzkirk. He's another Bay Area, long time turtle person. Very published, great character, good friend of mine. And I just reached out to him and asked him, like if he had any advice on where we should, what we should dig into. And he honestly gave us like one of the biggest next set of clues. And that's when he connected us with the paper written by Willie and Christoph Schneider. That was a 3-year study of them trying to essentially determine the extended range for Testudo climini in Libya. And in that, they actually, learned about, the extended range of climini into Western Libya. But in that paper, they make references throughout that entire paper that we found no climini in this area of Western Libya, but we found graeca. Right. And so far, to date, until we started doing what we were doing, it was just the assumption was that these are all an extension of the Tunisian tortoises. Right. So and Jim actually had photos and handwritten notes from Willie Schneider because they, had collaborated. And, like a lot of Jim's research was used in the Schneider's paper. And he showed me these photos, gave me their contact information, and Willie Schneider was the one that started giving us very specific information about, he had cataloged all of the details of their field study, and that gave us the biggest clues and the GPS coordinates for where we wanted to look. And, that really, really, sort of gave us a bit of the smoking gun, more of a smoking gun, I guess.
Nice. So you got the DNA results back. You kind of have an idea that you have a mystery tortoise, like Steve was saying, and then it clicks because you found a reference from 2017. And when you start thinking about, well, let's work together to define this species, because you couldn't find any other reference, what did that process look like, and how did you guys say, okay, here's what my role is going to be? What did that conversation look like?
Yeah, I think Steve kind of helped us set the, sort of the specific goals and the overarching structure, right? And then, I don't know if you want to speak to that, Steve.
And before you dive into that, Steve, when you even think of this, was anybody in the group saying, well, this sounds ridiculous, or no, it's just, was everybody pumped up to like, let's do this,
Mixture of both, I think.
Yeah.
I think we were
Fair
all
enough,
pumped.
right?
I think we were all pumped at the beginning
pumped, yeah.
because we had no idea how much work it was going to be and what we were getting ourselves into. So,
but some of it sounded preposterous, right? Like, oh, let's go, let's find someone to go do a field, do research and find one of these and get a DNA test. I mean, like, there's so many.
This was sort of like an impossible thing, right? We're
Right.
we're three people sitting in the U.S. and we're thinking, well, we have to find somebody in Libya.
Yeah.
That can go into the field in Western Libya, of course. And we've been corresponding with Murad in Eastern Libya. And of course, there's still, some difficulty within the country. It's not just, as simple as, drive to Western Libya and look for tortoises. So, I mean, I have to say that this whole effort, this paper, is a story in sort of the democratization of science as a whole. Like we live in an age, in an era where DNA testing is accessible enough.
Yeah.
And where you have, communication and where you have resources and you can find people to work with across the world on a project like this. And I think everybody brought something to the table. I mean, it's just,
I mean, for me, I knew what, my contribution wasn't going to be the details of the paper, because structurally I'm not really trained in any of that. Right. But I'd say how this kind of unfolded, though, like I don't know if it was by magic or what, but, we ended up getting, speculation started getting backfilled with factoids. Right. And every time we would get a new factoid, right, like whether the E1 designation, and when Steve dived into the the GenBank database, and we would kind of pick it apart, or he would pick it apart, and we would kind of collaborate in our chat, right? And then little by little, like every time we got a new factoid, right, or a new lead, I think it would kind of then ignite the fire for us to press on, right? And and little by little, that's kind of how it went.
Well,
I'm
that's what's
sitting here and kind of just trying to imagine the excitement when you talked about picking apart the data and trying to put. I mean, I think everybody here probably recognizes that at some level. If you guys go through a journey like this again, you got to set up a camera to capture your emotions and reactions. I'm sure that was just something.
We'll have to wait for the Hollywood movie, right?
And that's what I think is fascinating about the story, is that, to Steve's point, DNA sequencing is readily available, or it's pretty easy available, but it's, people get this idea, I'm going to provide this blood sample and you're going to tell me exactly where the animal came from. And that's like the furthest from the truth that it could be. It isn't magic. It isn't this, I'm going to provide this blood sample and you're going to put a drop on a pin drop on a map for me. And I always try to explain to people, there's got to be these arrays. You've got to be something to go back to and to watch, as you mentioned before, citizen scientists, discovering a new species. And a part of Libya, you could probably say has some civil unrest. It's hard to sample. I mean, you ran this across the finish line. It's just impressive that how it started and that it got completed. And I really do applaud the work here, because it really shows that, one, just as private individuals, we can contribute. Paul gets some animals, and I mean, I can't tell you how many times I've gotten a group of animals and said, well, this one's a little odd, I mean, you followed through, and your curiosity sparked an interest, and again, it's just such a great story, and one that you all should be very proud of. I mean, it is really, really cool how it unfolded.
Talking about different people coming together. So once now newbie question. I've never been part of some a description of a species or a subspecies, and I don't understand the significance of the authors being attached to an organization or an institution. So, James, if you don't mind me asking, Turtle and Tortoise Preservation Group is listed in the paper. What does that mean?
Well, for us as an institution, it's great to be tied to something like this. As a nonprofit, one of our as part of our charter is education, and part of that is our publications, and we see a lot of it on the kind of academic side with Bill McCord and his colleagues doing very similar work and identifying species, subspecies, and others who have, Jerry Fife and others doing their work with the Galapagos tortoises, which I've participated in, and others. And, it's as an organization, it's great to be a part of it, but it also is a demonstration that as privates, we can contribute, we can be a piece of this. And that's why I work in academia, but I see how important it is for citizen scientists to participate, and anybody can have an interest and participate. And this really demonstrated that. And for us as an organization, we're just proud of it, and the fact that we can in some way participate. But again, these are the folks that really did the work and really should be congratulated for the hard work and all the work that they did, and kept it going. So many people do what I do. Oh yeah, this one's odd, but it doesn't go any further than that. It took every one of these guys and their piece, their contribution, and getting it to what they got, which was a publication in Herp Review, which again, a very respected peer-reviewed journal, and their work, is looked at, is peer-reviewed and validated.
Yeah. Well, talking about the journal in itself, so you're going to publish this, has to be a peer-reviewed work. Did you knock on multiple doors? How did you get, or how did you guys land with this particular group?
I would say one of the things we looked for when we published this is a journal that would publish the article online, not behind a paywall, not with a fee structure. I think one of the things that was important for us when we published was to try to make sure that everybody could have access to this. And I know that's been a little bit of a contentious topic in the scientific community over time, and there's been more of a trend to favor open access journals. And I'm very much a believer in that. So no, to answer the question directly, we sort of looked for a journal we thought was appropriate for the work, and gave us a good chance of getting it accepted, and a good chance of and have it be somewhere open that people could see the work.
Nice. Now, what kind of surprises did you run into once you knocked on the door? I mean, I'm sure they gave you a bunch of framework and said, "This is how you got to fit this."
There's a review process. I think anybody who's published knows that the peer review process, exists. It is rigorous typically. In this case was no exception. We got a very good, constructive series of review comments from and we went through several rounds of revisions and improvements.
Lots
to the paper.
of editing.
And it makes the paper
You have
better.
a few battle scars.
It's a good thing, but it's a long process sometimes, and so we're thankful to go through that, but it does feel sometimes that we need to go change this again. We need to go make these updates. And
now, Oh, sorry, go on.
No, that's what I had. Go ahead.
Now, if I can go to Murad, a friend from Libya, you mentioned he's on the east side. Now the subspecies in question is on the west side. What did it look like at your end to organize volunteers to go look for this mystery tortoise and you probably haven't been in that region before, so you're kind of looking for something wildly out there in the wild. How did that look? What was that journey like?
Okay. First of all, this journey, it's not coming by chance. By the way, I worked in a project that talk about the protection some another species of tortoises, which is Egyptian one, Testudo. This one, it's in the red list in IUCN. So I worked that with many groups of Libya, but the problem was we didn't have database for everything in Libya. So for each species, this is good news. We got a new subspecies in Wisterian Bart. When I worked with my colleague, they told me we have to make a database that the scientific and the educational people who reach about information, they can access and get new information about the species in Libya. After that, I have groups with many people I know who work about tortoises. One of them, Mr. Nasser Bossin, as Iron and Paul mentioned, and they connect me with Paul, and so happy to be one of the team colleague that make these things it's come around. So the most important challenge, Libya, it's a big country. I think we have 1,800 kilometers in the coast. So to transportation between the Wisterian Bar to Wisterian Bar, it's so difficult. And also in Wisterian Bar, especially, we have some security problems. But for this work, I trust of my colleague, I trust of everyone. I have to do this one. I have to be the first expert in Libya. He talk about tortoises, so that's why I go ahead, and we got success finally. And as I mentioned before, I'm a PhD student, so I'm looking for to be to get more scientific papers to join with education, and also to join with scientific groups that's very successful for me. So that's why all of the group here and all of the people here, one of them, Aaron and Paul, and also Steve, now become the best friends in America. So I know both of them, they are in America yet, but no one visit Libya before. But I was like eye for them because I know the responsibility for this kind of work. I have to be more responsible to give them the good information to get good article and good research. Yes, sir.
I would just add, honestly, I thank Nasser Busen for introducing us to Murad, because without him, he made it seem very easy, like the enlistment of, this group of folks who ended up, going all the way across their country and, went into these regions. It's like, here's, let me throw a couple of GPS coordinates at you, and this is what it looks like, just go into these areas and see what you can do. And it was like they made it seem so easy. It was a few days, and suddenly it's like we found 7 tortoises, and it's like,
yeah. I mean, this was an interesting point in the review process. I think one of the early comments was, we didn't have a lot of detail on what the expedition was like for these tortoises. They said, "We'll talk more about how much effort, what areas did you search in something? And so we wrote back to the team in Libya to get these details, and we had no idea what effectively that we had asked for. I mean, this expedition was quite an undertaking. It was a whole group of people. They were searching many different sites over the course of a week. And so the amount of effort that went into that field study, even us, when it happened, we thought, oh, well, gosh, it's only been a week, and he's messaged us back and said they found tortoises. Oh, that's, we were thinking, oh, maybe they're really easy to find. And then later we find out, no, they mounted quite an expedition.
Sorry, Paul, you mentioned sending coordinates over to them. How did you guys land on that? What led you to those coordinates where you sent Murad and team to say, go look?
Yeah, so all of it is from Willie and Christoph Schneider's, field studies, and the fact that they took such copious notes and had all these handwritten notes, and they recorded almost every, location when they made reference to, like we found no Climeni here, but we did encounter graeca that looked like Nabulensis, and I said, do you actually know the GPS coordinates for that? And he shared it with us. He sent us extra pictures. We had additional pictures of them, and through his pictures, there was no doubt. I mean, because I think of the individuals on the call, I mean, like I had at one point many of these in my my turtle room, right? So when Willie sent me the photographs, I'm like, there's no doubt in my mind we're looking at the same tortoise. There were a few of them that were not, they weren't debatable, so at this point we just needed to really start going through and picking apart, all of the evidence and piecing it together.
Yeah. Wow. Yeah. No, I don't know if Murad is with us. I can't see his video, but I don't know if you can hear. How many days did it take for you and your team in Libya to track down these tortoises? If you can hear us. He might be having trouble. Oh, there you are.
Could you repeat again? I couldn't hear you as well.
Yeah, I was curious, how many days did it take, for you and your team to travel, look for these tortoises, and what were the challenges you were running into?
Look, about the days, I think Paul called me in the season of tortoises, which the tortoises be more active. I think it was in the spring in 2022. So we were so lucky because we got our samples in a few days. After that, before that, Steve and Paul talked to me many times. How can we reach the symbols as well? How can we take the swab symbols, and also how can I get the good symbols to get good result? So in the spring season, I think we got it in the few days. Yes, sir. But I need to say something. The hour now become to be 33 a.m. And also I have to go to my work early morning. So
I truly appreciate you hopping on. I think.
Honestly, Murad, thank you for joining us, and
thank you.
Like, it's been my pleasure to
شكرا كثير.
And I hope we, can do more work together.
العفو Iron.
Yeah.
Thank you so much.
Again, thank you so much.
Thank you.
Appreciate your work, and thanks for joining us. Have a great day.
Yeah.
Wow. So,
thank you so much,
Aaron or Steve. The samples that Murad and team collected, did those end up going for the DNA testing as well?
Yes.
Okay. Now, oh, sorry. Go on, Aaron.
Yes. It took quite a while to get those samples out of Libya, but they eventually were sampled. They arrived in Netherlands. They're analyzed, confirmed from the E1 haplogroup lineage.
Yeah.
And actually Stephen, through lots of, this was towards the tail end of the bioinformatics analysis, Stephen was able to locate something pretty interesting on, correct me if I'm wrong, one specimen. Isn't that correct, Stephen?
Yeah. So as we were doing the paper, as we were drafting the paper, and we had run all of the DNA results through programs to essentially read the chromatographs and give us the sequence. And a lot of these programs, they have some automated features to automatically select how to read the sequence. There's some adjustable parameters that you have to use. And, as we were going through and doing the analysis, we analyzed all the samples. We thought we did a decent job of pulling sequences. And all of the animals at the time, we really only recovered partial sequences for them. We got about half of the cytochrome B gene, which, some of the work we cited, some of the previous papers had managed to sequence the whole gene. So we're sitting here trying to figure out why, what did we do wrong? What did we I think we ended up with sample quality issues.
Mm-hmm.
We've talked to so many people about swabbing tortoises, and they're like, no, you gotta, it's a little circular motion and and so then we're trying to relay that information, but in the end of it, we ended up during the review process, we actually went back and reran the chromatographs to see if we could get any improvement in the sequences at all. And when we did that, we did find that one animal from the field study in Libya is actually a new haplotype. So prior to that, all of the haplotypes that we found were identical to the 2017 specimen. And with every single animal having the exact same haplotype, we were wondering, is this subspecies extremely genetically bottlenecked? Is there something, they're all giving us exactly the same mitochondrial DNA result. So, we were wondering if it was a very low diversity population, and there's no way to prove this from just a single gene on mitochondrial work, but we did eventually find the second haplotype. And so, there's some evidence that there is some genetic diversity in the population. We have no idea how big the population is. The range of this subspecies is unknown.
Mm-hmm.
It might be very, very small and localized in Western Libya, or it may stretch into southern Tunisia. We note that there's some of the purported Tunisian tortoises, nabulensis, in central and southern Tunisia look similar enough, and the habitat and the terrain is more similar to Libya than it is to northern Tunisia, where the classic habitat for the Tunisian tortoise actually gets quite a lot of rainfall. It's a very lush, vegetated environment versus, my background here is one of the habitat photos from Schneider in Libya, where it's basically very sparse, semi-desert grassland.
Yeah, I'd say one of the good pieces of information was, that they found these tortoises in the area that we got, sort of like these GPS coordinates from Willie Schneider. the Schneiders were there almost 20 years ago, right? So we were looking in this almost the exact places that they found these 20 years ago. So, I don't know that we've ever really talked about that. I don't know if that I've ever actually said that in front of, Steve, Aaron, or Morad, right? But if you really look back, I mean, the Schneiders did a 3-year field research back in like 2000, I want to say it was like 2005, 6, and 7, right? And here we are in 2020, I think it was 2023, the spring of 2023, kind of winter going into spring. But and they found these tortoises, not easily, but relatively in the same area. So I thought that was actually a bit of good news.
So what does that tell us about that region? Is it very sparsely populated, not touched by humans? What are we talking about?
No, some of the photographs that we got from the field study, we asked for, we collected pictures in some of the places where these tortoises were found, and you could see in the background of the photo there would be a multi-story high voltage power line or a house, or so strangely enough, we searched a lot of sites, and the tortoises were found in some sites that were rather disturbed. They were fairly close to human habitation, and the local habitat looked very similar to what we see like in my background photo, but these were not greatly far away from humans, and some of the other sites that we searched were more remote, and those sites we didn't find tortoises. So I don't think we know, although what we do know is, iNaturalist is a wonderful website, and that was a help in this study also, is looking at photographs people had published on iNaturalist of wild tortoises. And so from those photos, we sort of got a decent idea of where the range might be. Then that there's still a lot of open work. I mean, there is a need for some more field work, particularly in southern Tunisia, to locate the tortoises there and determine whether they are this new subspecies or they are Tunisian tortoises.
Yeah. I mean, it seems like also, like, if you look at like the papers, obviously I didn't comb, like a lot of the details as much as Steve did. So feel free to correct me if I'm wrong. But, even a lot of the DNA location data for Nabulensis is it's not widely distributed in Tunisia. Like a lot of those specimens were collected in a very small area, so a lot of the morphology that we kind of like just talk about, this is what a Tunisian looks like and things like that, is based on a very small set of Tunisian tortoises. And there are other sets of data that I think we call out, and there's a lot that is just not known about, like where, like across all of Tunisia, but particularly as you get into southern Tunisia and you get closer to the Libyan border, are those actually Tunisian tortoises that have been talked about for decades, or are these tripolitania, species, like, do those actually extend into Tunisia? And that's kind of one of the big questions that, we kind of pose in the paper.
Yeah, I think that brings up a point about identification. The Tunisian tortoise, especially Nabulensis, cannot be identified visually with very much accuracy. There's been a lot of historical, people post pictures of a Greek tortoise. What subspecies do I have? And we've had the tortoises that have been floating around in the U.S. for decades many of them have been misidentified. A lot of what people call Ibera is probably terrestris. What has been called Nabulensis in the U.S. is probably whitey. It's a mess. So Tunisians especially are variable. There's places where Nabulensis grows very large. So they're not universally a small tortoise even.
Yep.
Uh,
It's rarely mentioned like that one, like when we kind of looked at that, right? It's usually like that's the small tortoise. I think maybe because it was the only example of a small tortoise in the graeca, family of tortoises until now. But but the reality is there might be small whitey, there might be small nabulensis. Now we know that there's a Libyan tortoise that's also small.
There might be larger tripolitania tortoises as well.
Possibly.
Because if we're only sampling just a small amount, that people have smuggled out of Libya, and then the ones that have been studied in Libya by Murad and team, it's a tip of the iceberg.
Yeah, you can't exclude talking about the poaching as well, right? Because they're gonna that whole poaching route from Western Libya to Egypt, right, is a long haul. So they're gonna pick tortoises that travel well, right? So if it's a big one, let's say, they're not gonna probably load up, because some of these, Murad showed us lots of pictures, and some of these they're farmers that are just filling up sacks and carrying sacks of tortoises or filling a truck bed or something like that. And there's an optimum size. The Egyptian exporters know they can't be too small, right? Like if I'm going to send it to the U.S., there's the 4-inch rule, so they can't be too small. So we suspected, even like with some of those imports, we saw a lot of females and very few males. Like the male ratio was very low, and then it was our suspicion that the males are a little smaller. So they probably were hand-picking tortoises of a certain size, right? Yeah, for that reason.
Paul, I was so focused on the journey and the adventure you guys went on, I forgot to ask you to maybe send me some pictures of the subspecies and key identifying factors so we can share with folks. If it's not too late, if you have it on your laptop or PC, if you want to send me some messenger.
I can go grab one. I can send you pictures, yeah.
Well, I have one right behind me.
Yeah, you gotta go out of the way and show me the distinguishing factors there.
There's his head. There's his head. See?
Yeah.
There you go.
So and the size, since we're talking about, because I think one of our friends shared saying dwarf Greek tortoise, and I'm not sure if that will stick or if the idea is this subspecies is the smallest. What are we talking about? What can you tell us about the animal itself? And Paul, you're welcome to bring an animal and show if you want. Pictures you often translate better, but you're welcome to bring a live specimen if you like.
Just real quick, where do I send them? Like, where do you want me to just send it to?
If you want to send it to me on the messenger, that group chat we had, I can download it from there.
Yeah, sure. I can do that.
Yeah. So while you're busy with that, Aaron, what are your thoughts on that species, the size of this subspecies as adults?
Oh, I think it's a mystery still. I mean, there is so much ground to still cover, potentially hidden, unknown populations. Maybe there are other populations that are known, have been exploited already in the past. We have no idea about. Is there genetics gone from those, from those forever, or, I mean, when this was about 6 months ago or so, I was looking on Smithsonian Museum website, because that was part of my contribution, was to speak to museums to try to put specimens into their custody for the holotype, which is the definitive version, and then the paratypes of the deceased preserved animals. And so anyway, when I was looking on, I forget the name of the actual website, located within Smithsonian Museum, I believe between 2 and 3 specimens, maybe from the 1960s, that said Nabulensis out of Libya. And so I sent it to the guys and said, well, here we go, guys. Here are most likely, if it truly came out of Libya like it's labeled, these are tripolitania subspecies, and of course they were also fixed in formalin. So I don't know if any DNA will ever be able to be retrieved from them or not. But Yeah,
I think what Aaron's talking about, what Aaron's talking about, though, is kind of what we learned when we started looking at all this data, is that there's a lot written that isn't backed by DNA, evidence, and there is, I think it was Michael that kind of asked a question about, sort of, are there other studies on the graeca clade, I think is what he asked, and there are a significant number of studies that produced like all of this set of clade data that is part of GenBank, but when we started looking at it, particularly our curiosity about just the whole Tunisian comparison, right? We started realizing, because you can look at that data and you can see where the reference, the source of some of these specimens, and they were sampled all very close together in certain parts, right? So there's not a wide range, set of data for the Tunisian, and obviously, with this, I mean, there's so much more to be learned about, the actual population, and as you tunnel your way towards Tunisia, exactly where that boundary lies between these tortoises.
And also, I wanted to make one input in case anybody doesn't know. Nabolensis is the Tunisian tortoise. Nabolensis is the subspecies. Just in case you're not familiar with using Nabolensis instead of the Tunisian Greek.
I mean, at one point I even combed, because the UK has more of them, right? Historically, there were lots of exports of the Tunisian tortoises, from long ago. And there are a lot of, the survivors, right? And they've had success captive breeding the Nabulensis, and people post pictures of them all the time. There are plenty of Europeans that have very healthy, vibrant, producing populations. When you start looking at them, I combed through pictures, doing my obsessive thing, just to see if they all looked, like what the description of Tunisian tortoises was versus what I was seeing in these tortoises that I had. And, there are differences, right? But as Stephen says, when you really get down to it, you can find examples of tortoises in Tunisia that don't look anything like the Tunisian tortoise description, right? So there is a lot of variation that we don't know about, right? We've even seen some of it with the tripolitania. There are some that are, like with the Nabulensis, there are these light forms and dark.
The solid black one.
Yeah, well, there was that one too. Like in Nasser Busin is he's the admin of the Libyan Tortoise Forum, and one guy out in close to Tripoli, somewhere between Tripoli and Garion, found a dark specimen. Like it was almost, it clearly was, one of those freak specimens. It looked like, right, that it was what do they call it? Like melanistic, right? Mm-hmm. Like a melanistic version of this tortoise. But if you look at the structure of the head and things like that, it definitely looked like tripolitania. Right.
Now, before we dive into the pictures and the tortoise in itself, real quick, does anyone know? Sorry, Michael. By the way, good evening. Does anyone have any info for Michael or?
Well, I was just going to say, I think they touched on it a little bit, but there's a lot of holes in the genetic work because there's areas of civil unrest that haven't been sampled. And I also think early work made assumptions. So they would sample an area and then make the assumption for a larger area that this is the population. And so to Michael's point, Highfield and others, there's a lot of old records and stuff, but I really believe there was a lot of, there's what, I think it was Steve or maybe Paul mentioned it before. It really should be a dot on the map and not an assumption or a grayed out area saying this is the population. And I think a lot of that you had gone on at one time. And I know in the early 2000s when I was looking at a lot of that stuff, and when what we would call at that time Trestis or the Golden Greeks were coming in, and the Black Greeks and all of that, and we were all looking at different ranges and stuff like that, and a lot of assumptions were being made, and that's why I know Andrew Hermes, when he had he had some DNA work done on some of his, I was like, where was this done? Who has the panels, because it always seems like people just want to run off phenotypes and I think Aaron's mentioned it, or maybe they all have mentioned it now. There's a lot of locale variance, and you really need to have that DNA to back this up. So that's again what made this the gravity of this work so important and valuable.
So, thank you, James.
There's a baseline. Oh,
oops,
we've got some
little bit of feedback.
There's a baseline of data on Testudo graeca, though, that we used, obviously, for this, right? I think what we just learned is, there's just it doesn't go very deep, right? It was very broad, and the clades were, sort of mapped, and it's very difficult to map the clades to morphological characteristics and be confident of what you have, right? Is I think Stephen's point. I mean, without a doubt, there's probably a median where, most of the nabulensis might look like this, right? But there's always, I think graeca across the board are like this, right? You can see a terrestris that looks like it's from North Africa, but if you don't use multiple criteria and just start eliminating and saying that, North African, graeca usually have these characteristics, and you go through your list, it has to qualify for many of these characteristics before you can feel like maybe, you're correct, right? But, so far it feels like DNA is really the only, way to truthfully know that you have, one or the other. And, and then, like I've said many times, it just seems like there's a lot of record, but it's just based on very few specimens, right? Like some of them are sampled really in high volumes, others fairly low volume.
And it comes down to, I think, people in the hobby jumping in and doing that, right? The test. So James, you're probably close to this, but Steve or others also from your journey might have this answer. How close are we to having a DNA testing lab here in the US that people can very easily access, send samples to, and get an answer, actually, so that,
That's an absolutely wonderful question, Michael. I love it. Who wants to start one?
Yeah.
Well, I think that's the other thing. I think based on the answer, hey, here's a business opportunity, maybe. I think
there's a need for this. I think having a PCR lab or a DNA lab that is serving anyone, equal opportunity, private sector versus public within the U.S. would one, be extremely valuable for private sector conservation, where we don't have easy access to university facilities. And it would really enable a lot more citizen science. I mean, there's a lot of mysteries. I mean, I wish somebody would have, we don't know if those giant yellowfoots are genetically different than normal yellowfoots. We don't know if the tiny musk turtles in one part of Florida are different from normal sized musk turtles. I mean, you could come up with a half a dozen mysteries in the tortoise world right now. Leopard tortoises probably consist of 6 or 7 subspecies. They just, nobody has been able to put together enough work in one place to clean it up and delineate what they actually are and what the ranges are. Red-footed tortoises might actually have subspecies within them. Same problem, though. So there's an enormous amount of work to be done, and it's not hard to think of things where studies need to be done or papers need to be written.
I mean, it's a huge barrier, right? Like for anybody who even has something that they want to chase, to do what we had to do, right? I think I've walked, I had to teach myself, I'd never taken a sample before, like a DNA sample, so I'm, looking up on the internet and asking other folks that have done it before, just to learn about it. So it wasn't a big surprise that my samples weren't the best, then we had to sort of train, these guys that were just gonna, embark on this, travel from from Eastern Libya to Western Libya, so that if they happen to find tortoises, this is what we're going to ask you to do, right? Like, stop what you're doing, take a bunch of pictures that look like this, take these measurements, and then try to get, samples of these types using this method. I think at one point I filmed a video and sent it to Murad so that he could share it with the team. And to be honest, like, if it weren't for having access to Gendica, I mean, honestly, it just wouldn't have happened, right? Like, the fact that Gendica does this for anyone, right? They do a lot of work, they do a lot of other scientific work, and we were very fortunate. It takes a long time. You have to not only learn how to do the sampling, but you have to learn how to package it up so that no one confiscates it, leaving the U.S. and then entering all these other places, or gets confiscated at the Netherlands border. And then, by the time it gets there, hopefully, these samples don't fail. Luckily, we didn't have one single sample fail. They weren't they weren't complete, but I had one sample fail, but it wasn't these tortoises. I sent also some other tortoises just to see, I was like, let's see if I can get the correct turtle species detected. Because
was it a Cyrenaica, or was it a
no, it was actually one of my Dalmatian Hermans, who's just
okay.
This beast. She's like 8.5 inches. She doesn't match anything, but it was she came to me from a very reputable person. And I was like, I just want to see if it comes back as Dalmatian, because she's so huge, but she's got all these other characteristics. That one sample failed, so I never got the answer on her, right? But of all the Libyan tortoises, all of the ones that look like Cyrenaica came out in the C clade, and all of the ones that looked different came out in the E clade, right?
Sometimes you catch breaks. It actually works out.
Yeah, we were very nervous of putting all this work in, that we'd send the data samples and we might not get good data.
Yeah, and we were nervous.
Part of the journey, right? Yeah.
When the samples left Libya, we were so nervous about them that we actually had Murad split them into 2 different shipments, because we were afraid they were going to get lost, right? So they sent the first one.
That's a great call, yeah.
And then sent the second one after. Yeah.
There also was, if I'm not mistaken, during that time period, the hurricane, I believe, that had occurred, and then tons of flooding. What was it? A dam break?
Yeah, Murad's town was almost washed away. Darna, Libya. I don't know if you followed when the floods actually happened. They had like I think they called. I forget. I mean, we call them hurricanes, typhoons, like
very catastrophic. Essentially, a hurricane in the Mediterranean hitting Libya, a place that gets no rain, getting just completely inundated and trashed.
Sidebar: they had tortoises because all of these, we haven't talked about this, but with the Cyrenaica tortoises, and also these, they come from very high elevations. They're usually like 2,500 feet above sea level. So they're on these like higher, it's almost like high desert kind of areas that are, more lush and green by Libyan standards. So when all of this rain happened, they had tortoises that washed down into Darna, which is closer to the coast. And they were finding tortoises in the mud that they were rescuing, that were coming from like the higher elevations from all the floodwaters.
I've read of similar events in papers that were describing some Asian species and stuff. So yeah. Wow. James, didn't mean to cut you off for any comment you might have had about a DNA sampling facility here before I dive into these pictures.
Oh, not at all. I mean, I think Steve answered the question very well, and I mean, there are labs here, of course, but having access to labs to not only do the DNA work, but also that has the reference panels. I get this question asked to me all the time. Where can I send it to DNA so I can find out where my tortoise is from? And I'm like, it's going to tell you a piece of the puzzle. It's not going to pin drop for you, and I think people need to really understand that, because you guys did a lot of work, and the DNA portion was just a piece of that puzzle, and you had to really do a lot of work to complete this picture. I just didn't want that getting lost in the messaging that, oh, I'm just going to run a sample and it's going to get, I'm going to get this magic pin drop and I'm going to know exactly where my animals came from and I'm just going to do all of them. If it was that easy, we would all have done it, right? So.
Well, thanks for the pictures, Paul. Here,
Sure.
Let me dive into that. Beautiful animal indeed. Now, how many of these have you seen so far?
So like when they first started coming in and we started realizing they we thought that they were unique, and I started posting online, other people started reaching out to me after they saw them, who individuals that have become good friends that ended up buying as many as they could see, and we ended up then with at one point it was like 35 or so of these tortoises, and then split them up into different groups. But we had mortality, of course, right? So these are all photos of ones I have now that I took shortly before they came in for the winter. But their mortality rate was actually fairly high. I mean, like the Libyan imports in total, the mortality rates were very high. They were well into the 60 percentile. And with these, I'd be surprised if it wasn't above 70%. They are an extremely sensitive tortoise to change. I think, like James was describing, another tortoise that was, sensitive to big changes. And it seems like these are definitely, like once you have them in a good place, you don't want to move them. They respond poorly to just going out for the summer and coming back in for the winter, right? Because I can't keep them outdoors full time, and they always go through this sort of thing. Obviously, mycoplasma is probably omnipresent with a lot of these tortoises, so it's I've learned a lot.
So between the 5 you picked up, or 2.4, I think you said, sorry, 6 you picked up and 3 were different. What were the telltale, or what are the telltale signs for this particular subspecies, if there are any that you can point out to us?
Yeah, I mean, like early on, obviously we didn't get to the point where we were kind of picking it apart, like microscopically, like we did in the paper, right? But early on, when they came in, they were all relatively the same size, right? And of course, I was, I had been looking at Cyrenaica for a while, so you're used to seeing all the speckling and marbling and like the nice fenestrations, and they always have like lots of different patterns and things like that, right? But usually not very uniform. These were very uniform, right? Like if you look across them, I mean, there's some variation. Like you can look at the little male that you showed at the very beginning. He's a little lighter, but for the most part, they're uniform, right? They kind of have, sort of, like across the marginals, they have kind of similar patterns and things like that. With the Cyrenaica, they're not like that at all, right? Like they're just all over the place. Like they just don't have any of that uniformity, right? And these were far more domed than the Cyrenaica. Cyrenaica, they tend to be elongated. Some of them they're all very domed tortoises, but if you held them side by side, these are very short, compact little tortoises. They're like, the females are like little baseballs, and the Cyrenaica obviously start to get flaring in their marginals, and to me, the Cyrenaica Libyans have a look like it's their brand, like they have
we call them a bell shape.
You usually know you're looking at one, right? And there are exceptions, but when you look at these, they just kind of look very uniform.
We used to call it a bell shape we called it a bell shape in the Syrians or Cyrenaikas. Because of that flaring, you would say it was a bell shape or a Greek. That's how we would describe them early on.
And because of the sheer number of Cyrenaica that came in from 2021 forward, I mean, like honestly, it felt, well, at one point we actually could see like some of the import data, and our estimates are like probably 2,000, Cyrenaica Libyans that came in. And because we got to see so many different specimens, there was variation. There were some that were a lot more domed. I had them tested and they came out as C. There were some that were very oblong, and had less flaring. Those were tested; they came back as C. But if you look at the C clade, there's a lot more of the different. There's a C1, C2, C3. I forget exactly how many there are, but we couldn't quite tie a morphological pattern to to a particular clade. But with these, they were more uniform. We saw some variation, but there was no one that would confuse them, right? Like if you were looking at them, you wouldn't confuse the two.
That's good to know.
Anyway.
Sorry, say that again.
I wonder if they were coming in. I wonder if they were coming in the earlier shipments from the like 2006 when when I first started seeing the Syrians come in and stuff, or the Libyans. Sorry, not the Syrians, the Libyan Greeks.
I'll say this: when I deep dove into like the Egyptian forums, which is where I learned about who the exporters were and sort of like the trafficking part of this, because I was looking for pictures. I just wanted to pattern match to like these. I learned that these were very hard to get for the Egyptians, right? And if you look at it geographically, and you start talking about the political strife in the countries themselves, like the effort it took for Murad and Ahmed to actually go from Eastern to Western, in their own country. The poaching and trafficking lines are much more difficult going from Western Libya to Egypt, obviously, right? So does that mean in 2006 it was impossible? Maybe, could there have been some? Yes.
Interesting.
Yeah. Well, and that's where when people ask even about some of our North American species, they're like, are these pure? I'm like, well, if you know the history of box turtles, for example, how between clubs and stuff, how these animals kind of cohabited in backyard and stuff without doing a DNA testing, it's hard to say, oh yeah, this is a pure animal. It looks visually pure. Something I tell people even at this day and age, and they probably think I'm being stupid. I don't know. Paul, so you guys started this journey in 2021, and you kind of, in parallel of being on a different journey as well, kind of tracking down these animals here locally in the United States and kind of connecting keepers. What does that look like today? What does the status of this subspecies in the US look like today? What's your thought?
I think I was doing some cocktail napkin math with the guys, like a couple, maybe it was last week before this, and there have been far fewer of them, like of the big Libyan imports, right? The majority of it have been Cyrenaica, and in those imports there were like some of these, and then since then there have been some imports of just these. It's probably pretty low. I mean, like it seems like at this point it's definitely under 100 animals, across probably like 7 to 9 different keepers' hands, right? It's a fairly low number, and the male ratio is still fairly low, right? At one point, I think there was a concerted effort to try to get some more males, and some more males were actually obtained, but for whatever reason, we don't understand why the mortality of the males is actually higher, at least in our experience. And obviously we don't have a lot of numbers to sort of back it up, but the males seem to struggle more than the females in captivity.
Wow.
That was true. That was true even for the Libyans from 2006, and that everyone was looking for males, and managing them was very hard. Everyone was looking for males.
Yeah, so many patterns that match.
Yeah.
Now you mentioned you bring them in for the winter. Is that data driven by where they are found in the wild and what the conditions are like, or are you just not comfortable enough yet with this group?
So in my deep dives, I also like monitored the weather and just really tried to understand sort of like some of these regions. And at first it was like speculative that they were from this Western region, right? But just in looking at, you know, where I am in Sonoma County, I'm coastal, but I'm like, I'm inland a little bit, but I've got this San Francisco Bay influence, right? So I end up getting a lot wetter winters than most Californians. And you know, like the amount of precipitation in these regions is a lot higher than you would think, right? Like it's not like, you know, like a lot of people are, you know, would kind of just look at a map and just look at a few pictures and say it's Libya, it's desert, you know. But in these regions where these tortoises are found, it is the green belts, and they call, you know, the Cyrenaica Mountains are called the Green Mountains just as a common name. And then Jabal Nafusa, which is, you know, the Western Mountains, is also kind of a green belt, not quite as green, you know, but they do get like fog influences and things like that. Here in Sonoma, I knew my winters were a lot colder, right? Like I mean, in a snap freeze, I can get well into the 20s, and in a really bad year, I can get into the high teens, right? So I never felt comfortable taking the chance. Maybe if I had like the right mix of greenhouse and sheltering, I just haven't really kind of found that, and also, you know, just didn't really want to risk it.
Makes sense.
I think the other factor in play here is that all of these imports have, at a minimum, probably mycoplasma with who knows what else. So, you know, there's an interesting topic when you start talking about the native climate of these North African graeca, because North Africa in particular has seen extremely wild climate swings in the recent geologic past. So, you know, since the Pleistocene era, they've swung back and forth between sort of very dry and very wet climates. You know, in the ice ages, things were obviously much cooler. So there are other times when North Africa would have been much cooler and much wetter than it is today. And so, you know, I think there's certainly that is important for people to understand as they're trying to understand what their tortoises might be able to handle.
Mm-hmm.
It's not out of the question that they could handle wet weather and cooler weather, especially being from more mountainous regions. But I think the problem is there's very few. There's a really big need to try to conserve the ones that are in the U.S. and to try to at least get some sort of a captive breeding program going or something. Since, I mean, they're already here. You know, we certainly don't want to see more exploitation of these in the wild, given the circumstances. But we also don't want to lose the ones that are already here and just have them sort of fade away and all die off without leaving any successful breeding behind.
And just to insert from there, Paul has gone through days upon days upon days of ICU type care of his tortoises, from nebulizers to force feeding, all different types of medications. He's tried everything. He put in all the good work. If the animals that did perish die, you know, the ones that did perish, it wasn't anything to do with the effort that Paul put into it, because he has he's done the, you know, the groundwork for it.
And several others have as well. In fact, I've had some conversations with some Europeans recently, because Europe has started to see Libyan tortoises in very small numbers. Europe has a lot more regulation around it, right? And there are people that are somehow skirting regulation, getting small numbers of Libyan tortoises, and they are getting into some good keepers' hands, and they're learning a lot too. I mean, it's pretty much the same experience where, you know, a lot of these, you know, from the point of collection through all of the different points of, you know, of facilities and exportation to their country of import. I mean, these tortoises go through a lot, and many of them, when they land, it's amazing that there are as many as there is still alive, right? Because of the journey that they've undertaken. But I learned just recently some veterinary work was done on some Cyrenaica libyans, and including an endoscope to check what the organs looked like on a live tortoise that wasn't doing well, that was perceived to be lost pretty soon, and their internal organs were considered shrunken, right? So they had been, even though they looked outwardly healthy, they looked hydrated and things like that, they clearly had other underlying signs that that they were in a very diminished state and probably weren't going to survive anyway.
Yeah. I was going to go down a memory lane there. When my early experiences with hinchbacks, I would read about. Oops. I'm getting feedback. I would hear about or read about how animals looked healthy, looked like they were gaining weight, and then they just crashed, and necropsy would show that the organs were, swollen and whatnot, So
you always have liver failure too.
Yeah,
that's one of the things that you almost find it's ubiquitous.
And it was kind of you to point out the work that Paul has put in, Aaron, and I know you've done that with those hingebacks, and and I think that's the beauty with many of the dedicated keepers. We heard at the last conference the effort that Doughton and his wife put in managing Tink and their Galapagos tortoises. I mean, it's an incredible story, fighting to save these animals. And again, it's sad when keepers get lumped in and thrown into all in one bucket. It just diminishes the work that people like you, Paul, James, Steve. I've talked to Steve at one point. I was shipping some animals, and his focus and care in even the kind of box I was using, whether it was reused with some other animals. I'm just not kidding here. The kind of thought that many of the keepers put in when it comes to biosecurity and stuff, it's no joke, and it's a shame that doesn't get recognized. Again, thanks to some friends who make a lot of unnecessary noise as well. But kind of, keeping in mind that it's getting late, I know James has to drop off. A positive note, where do you gentlemen go next? I mean, you've been on an amazing journey. You've made some amazing friends, lifelong friends. What is next for each one of you? What has this journey kind of inspired you to do? Paul, we'll start with you.
Okay. Honestly, I really don't know. I kind of, once this paper was published, it kind of was published a little bit of a surprise. Like there was one day where we were just kind of, I think someone just started posting on Facebook about it, and we're like, oh my God, it happened. But we didn't know, we weren't notified, and I think once it's out there and we knew there would be interest in talking about these things, I personally want to focus on, sort of the captive breeding, look at, potentially a stud book program. We've kind of been, talking about, that with some of the keepers, and, just doing whatever we can to establish, a viable path forward, with captive breeding them. And then I want to do as much as I can to enable Murad, and hopefully, if there are opportunities for us to do additional work, that he, is in a position to be able to lead it there. If I ever had the opportunity, I would take it, but from what I understand, that's not always, like a safe and possible thing to do right now. But, I would love to see, some continued research, from where our paper leaves off, and start filling in some of the gaps that we don't know.
Absolutely. Awesome. Aaron, you've been quiet, so I'm going to go to you next. What do you have?
Oh, well, there is always something. There's always. I'm hearing feedback.
Yeah.
Is this messing up? I'm wondering now too.
I don't know. Steve, can I ask you to mute yourself if you don't mind? I'm going to mute myself. I don't know if the speaker loopback is getting picked up. Yeah.
Yeah. Can you hear me better now?
We can hear you.
Okay, you can hear me. Okay, good. I haven't ever seen these tortoises in real life, so that would be one thing that I'd like to see. One step is actually see the tortoises that we've been talking about for 3 years, and beyond that, captive breeding, facilitate about biosecurity to keep everything that is there alive and healthy. And, you know, if there is a publication that any of us can work on that we feel that would be valuable to bring, again, the academic community, the private community, public community all together, that's something that we would be definitely interested in. And that could be with the tripolitanian tortoise. It could be another species that we don't even we haven't considered yet.
Yeah. Well, I'll make a shameless plug in real quick. Hearing feedback. Sorry. Aaron, if you don't mind, can you mute yourself? Here, I can do that. Let me see if that fixes it. Okay. Yeah. I'll make a quick shameless plug in. You know, feel free to write an article for us on the subspecies in this upcoming year's Batagur. There you go. You know, share with the keepers, and Paul, I'll reach out to you about that. Sure. Steve, how about you?
Yeah, we talked about this, Michael. We know we all owe you articles, so I think that I really think that we need to collectively find ways to do more private sector conservation, more meaningful private sector conservation. And I think we have to sort of do it in a way that bridges the gap between us and the academic community in a way that, you know, where it gets seen. I mean, we have to publish in mainstream scientific journals. We have to go give talks at the conferences where all of the researchers are. You know, it's nice that we have our conference, but we need to go where people, other people hear us. And, you know, I think in Europe, there's a lot more active and continuous cooperation between, you know, private sector groups and zoos, and you know, that there's not as much of a, you know, it's a lot easier to work across this gap in some places. And I'd like to see it become more like that in the US. So I don't know where that necessarily leads me specifically. But you know,
I'll encourage you to keep poking. You know, that's when movement happens. And you know, talking about Europe, haven't had the privilege of going over there and observing. I don't know how much of that has to do with the attitude, right? I want to say a lot. You know, the collaboration. I think attitude has a lot to do with it. Of course, I'm doing only the TTPG conference. We're seeing a lot of folks from zoo facilities come and attend, learn, share. I want to make sure that I point that out, that they are sharing with us. You know, that learning goes both ways, and I think I can, and James knows this, I can confidently say we're going to continue to, from TTPG standpoint, continue to work and extending that invitation and continue to work on facilitating that collaboration. You know, so again, keep poking. I'll leave you with that. Excited for the future, man. Thank you. James, anything before we bring this to a close? Thank you so much for making time. I know you're staying back after work and making this happen.
Yeah, not a problem at all. And I completely enjoyed it and enjoyed the conversation. And again, it's been an absolute privilege to spend the time with these guys and hear the story. And you know, it really will make those connections that I think that Steve's looking for, and others. I mean, this is how you bridge those gaps, and looking forward to some more good work here. So, but my next project is the conference, so we're going to be starting to put it together for that, and looking forward to another great year. So, and I think this article or your publication would be good in the monograph, which is on TTPG's more scientific journal, and when we put together that next issue, I'm sure Russ will be reaching out to you guys to inquire about whether you'd want to publish in there as well. But again, it's appreciate everything you guys have done.
Yeah, thank you, James.
Thank you.
And talking about the conference real quick, this is going to be our 15th annual conference. I mean, 2024 would have been, but we all had to go through COVID. So I hope you gentlemen can make it. I hope those of you who are listening and watching it, can make it. Paul, looking forward to saying hi in person. That might happen this year. And Aaron, same thing. I'm looking forward to meeting you in person.
Absolutely.
Steve, I hope our paths cross again this year. If not in TTPG, maybe somewhere else.
Yeah, I've got plenty of people that twist my arm every year. So y'all should do it East Coast, West Coast thing.
Let's not go there, man. Let's not do that. Be nice to James. James and Drew, they make the conference happen, and that's why it is there. And Arizona is nice that time of the year. I'll tell you that it's
good weather for that.
Yeah. But again, gentlemen, thank you so much for your time. Thanks for sharing your story with us, and I hope your story, your journey inspires other people. I hope it inspires other people who have been sitting and saying, hang on a second, there's something unique about this animal. I hope it encourages them to connect with others. Again, I think we've shared this on this platform. We all do more for these animals by working together, and that's something I hope we can continue to keep doing. Thanks for your time.
Thanks for having us.
And thanks for watching.
Thank you, guys.
Yeah.
Our pleasure. Thank you. Have a nice evening.
And great to meet you both as well. I'm looking forward to continuing to get to know you. Have a great evening.
Likewise, Paul. Thank you.
All right.